Well, synthetic blood approaches include oxygen-carrying solutions and the cloning and growth of haemopoietic stem cells to produce more blood cells, specifically red blood cells. There's an article just recently come out in the journal or the paper for the International Society for Blood Transfusion, which actually talks about a project that's happening in the United Kingdom at the moment, cloning red cells for artificial, inverted commas, blood use. So it's been a bit of a holy grail for a number of people. The problem is essentially that the problem is that people are better manufacturers than artificially done in the laboratory. Well, for the oxygen-carrying solutions, the potential benefits include a long shelf life for the solution. So at the moment, red cells have a 42-day shelf life. Platelets only have 5 days. Fresh frozen plasma is 12 months and cryoprecipitate is also 12 months, but that has to be stored below -25. So red cells, of course, are in a blood fridge, which is between 4 and 6 degrees, and platelets have to be between 20 and 24 degrees on a shaker to keep those little cells alive. So artificial haemoglobin carrying solutions have appeal because if you're in a remote place or wherever and you can just grab it off the shelf and use it, or in a war zone or something like that, then there's a big plus for that. But for the artificially grown or the universal donor cells or the particular niche type of red cells that have been cultured, it would mean that blood may not have to be cross-matched, or that you wouldn't actually require volunteer blood donors. And that would save a lot of cost and a lot of time as well. Well, oxygen-carrying solutions are often toxic, and they don't always release the oxygen to the tissues at the right time. So the red cells are designed to carry the haemoglobin molecule, which is the complex protein that binds oxygen at a low at a high oxygen concentration and then releases it at a low oxygen concentration, which is in the tissues. Haemoglobin without the red cell envelope is toxic and damages tissues and has to be mopped up and sequestered, and it's dangerous. So the body has got methods to actually mop it up and get rid of it. So there's two things. So they're toxic, and often the oxygen dissociation curve is such that it doesn't deliver the oxygen to the tissues at the right time or the right place. And then in terms of the culture of the red cells, is that people really are the best manufacturers of red cells. We've been millions of years of evolution have gone into us being able to make them, and make them well and quickly and efficiently. But upscaling production for the cultured red cells has proved very difficult and expensive so far. You can make small quantities, but in terms of the immortalised cell lines that they've got to produce these red blood cells, some of them get worn out after a period of time, so that there's only a finite number of times that they can actually make the progenitor cells and then make the red cells. Others are immortalised and can go on for a long period of time, but they are not very good at removing the nucleus out of the manufactured red cell, and there's other things that just aren't right about them. So there's still a long way to go for those things. Well, I would still say that it's a long way off in terms of replacing volunteer blood donors. The work that's going on currently in the United Kingdom is looking at more niche products for the untransfusible patient who's either got a very rare red cell phenotype or very rare antibodies to high incidence antigens, so therefore there aren't any compatible donors. And there are people in the world for whom there might be just one other person who is a match in terms of their red cell phenotype, or there are some really rare ones. And certainly I've dealt with some of them here too. We have them in Australia. But essentially, in terms of artificial blood transfusions, we still need to fully understand all the processes involved in the production of red cells in humans, including all the factors that in play in the bone marrow where blood cells are produced. Biology is really complex, and there's still lots of things we don't know in the biochemicals pathways, in the environment in which red cells are grown and are able to mature, and all of those sorts of things. There's still lots of questions.